All right, good evening. We are going to have three videos this week, although one of them should be fairly short, this first one. And then we'll have a sort of special video, and then we'll have our journal club, which will also be kind of short. We had a long lecture about SARS-CoV-2 and COVID-19, and now we want to go through and we want to pick up where we left off with the last thing, finish up a few more things that we want to put in that class lecture category, and then we'll move on to two other kinds of videos. So, the thing that you want to realize when looking at all this, we talked about the epitopes that T cells can see and how some of those epitopes can be shared across coronaviruses. There's about, I think it's like 33% identity and maybe 50% similarity. I forget the numbers, but they're substantial, but not everything. So it makes sense that you would have some T cells that were ready from the common cold exposures, but they wouldn't be everything. The good news is once you get the T cell immunity, after everyone talks about antibodies all the time, I want to talk about T cells because the advantage that T cell has, it takes longer to kick in, it's more mild at first, but it has a broader base of reaction. That's why it can be so broad that you can have some T cells that respond from previous cold coronaviruses. Because we're only talking about peptides bound to MHC, little short snippets, and those can happen to be the same for a lot of different things. There's only a few parts of those that are really presented to the T cells. That means the chances of them being the same are higher. Because of the cross-reactivity chance, there's also the chance that the virus is going to make a few mutations in one of the peptides. But remember, you have 30 to 40 peptides that your T cells respond to. So maybe they make a mutation that's going to destroy one of those. Oh no, you're down to 29. Again, this broader base is what we mean. It's easier for a virus to mutate and evade antibodies than it is to evade T cell immunity, just because there's so many more T cell epitopes, and only there's a few locations in each that are crucial to recognition, and there's just a broader base. There's more epitopes is one of the ways to look at it. So in fact, here's another thing. People who've been infected with SARS-CoV-2 typically generate T cells to at least 15 different fragments of coronavirus proteins. Remember that was the graphs that we showed you, 4 to 5 for one kind and 3 to 4 for the other kind. And that's only a fraction of the possible. That only shows you what proteins you respond to. You usually are responding to multiple fragments from each protein. And so depending on how you look at it, it can be 15 different peptides that you respond to or 30 to 40 as the previous thing said. More than a dozen, that's for sure. It makes it very hard, and this is Alessandro Setti himself, this makes it very hard for the virus to mutate to escape cell recognition, says Setti, unlike the situation for antibodies. And so remember that cross-reactivity can happen from B cell epitopes too, and there's studies about that. Setti is a T cell scientist, so he doesn't study these, but there's other groups that study cross-reactivity. And there is some cross-reactivity at the B cell level. You have antibodies that can be cross-reactive. In fact, some people are looking for a super cross-reactive antibody that would be a monoclonal that we could give that the virus would find it very hard to mutate away from, sort of a universal coronavirus blocker. And there's some claims about that they might be closing in on it. I'll believe it when I see it. But the main thing is, this can happen for a B cell epitope. It's just a little less likely for B cell epitopes, and you have fewer B cell epitopes that you respond to. But here's a study looking at the extent of cross-reactivity. This is antibodies that you develop to different proteins on the COVID-19 surface. And notice how you have all the proteins again from top to bottom, and you have people with mild COVID in the middle, severe COVID on the left, pre-COVID on the right. And so you see on the left, the severe COVID people have a lot of blue dots right next to the S protein. And also look down there, they have a lot of blue dots next to the N protein. Not really many blue dots to other things, but the question is, okay, you've developed all these antibodies against the S protein and maybe a few against the N protein. How much do they cross-react with, say, the common cold strains? And they went and they measured it. So they went and these are the B cell epitopes that they're showing right here. And they're showing that some of them are cross-reactive. The one on the top is cross-reactive, the one on the bottom, and we're looking on the right here. The one on the bottom is not as cross-reactive. And so that's what they measured. And this is the main thing that they looked at. And they have a tree of all the different coronaviruses. Like we said, there's the 4 common cold coronaviruses. Those are scattered throughout this tree. Then there's SARS-CoV-2 and SARS-CoV-1 is here. It's a little bit hard for you to see exactly what's what. Under the orange beta petal of the tree, that branch has SARS-CoV-2 and SARS-CoV-1, and also the bat coronaviruses that look like they might be intermediates between SARS-CoV-1 and SARS-CoV-2. The common cold viruses are actually on the blue and blue-green, the branches pointing up, but they're still on the same tree. They're more distantly related. They're cousins and not siblings. SARS-CoV-1 and 2 are siblings. They're brother and sister. But the cousins are up there on the top. But because of that, there are some antibodies that you form against spike after you've had COVID-19 that will also react with SARS-CoV and with the other things as well. And so they have this chart with SARS-CoV on the top. You see all the blue. That means the different patients, 232 patients, have good antibodies against SARS-CoV-2. Right underneath it is SARS-CoV, and you can see there's still a lot of blue there. That means there's a lot of antibody binding to SARS-CoV spike. You move down to MERS, and the MERS is the third row, and it really has zero, not zero, but it's much more light in blue. So that means that MERS is more distantly related. It's actually on the bright green branch, the one that's sort of going to the side, on to the left. And so that means that you actually don't have much cross-reactivity there. But you move down to the bat coronaviruses. Again, these are siblings. These are on the orange branch. And stuff on the orange branch tends to have antibodies to other things on the orange branch. So antibodies are a little more focused, but still cross-reactive. And you can really get into this if you're a virologist. But for us right now, we just want to say this is where we see the cross-reactivity. T cells are more cross-reactive than B cells. But of course, B cells have lots of advantages, in which if you have an antibody, you have something that literally blocks viral entry in many cases. So the other thing that we want to talk about is, remember we talked about how cross-reactivity is where you have the common cold and maybe you have some T cells from the common cold that will cross-react with SARS-CoV-2. But remember, there's also a downside of cross-reactivity. Cross-reactivity between a virus and the self, if the proteins look too much alike, and if you get the protein from the virus defined as bad, and then you have a self protein that looks like that protein from the virus, that's what you get autoimmunity from. We talked about that as molecular mimicry. And there's a case where we've seen molecular mimicry that explains one of the worst aspects of SARS-CoV-2 for kids. It's not SARS-CoV-2 infection that much for kids, but it actually was associated with this syndrome, multi-system inflammatory syndrome. MISC is what it was called. And this was observed in children after they had been infected with SARS. They had an inflammatory reaction that appeared to be autoimmune. It turns out that it really is autoimmune, and in fact, we have located the exact peptide that gets activated against in your self protein. And this peptide does look a lot like a SARS-CoV-2 peptide. And so this is the case where the infection is like what we had the quiz question on. Here's on the top, they have the self-peptide that's being reacted to in pink. On the bottom, they have the, actually, I hope I have this right. I need to look back and forth. Oh, yeah. So actually, I'm not sure which one of these is self and which one of these is the other one, but it doesn't really matter, because what matters is that there's this epitope right in the middle of the protein sequence. They tested it by mutating them to all alanines. So like mutating each section to all alanines and then seeing how that affects how antibodies are developed to that. And so whether the pink one's on the top or the bottom one is self, one of them is virus and the other one is self. I think SNX8 is virus and then MADS is self. But it actually works either way. What matters is you get one of these first, and they overlap enough. If you look at the letters have a reasonable similarity. They have that PQG, and they both have that. And that's apparently prominently exposed in the epitope. And it looks like it's the right length for a T cell epitope, although I'm even not 100% sure of that. The main thing of this is some kind of molecular activity where you have a virus, the virus infects you, gives you one of these sequences, and then it reacts against the other sequence, which is a normal self-peptide. Then the children develop MIS-C from that. The good news is now that we know it, we know exactly where the MIS-C is coming from, and we can try to combat it. But again, autoimmunity is one of the hardest things to combat because it's taking a good thing, your immune response, and turning it against yourself. You want to be able to shut off that one part of the good thing, the immune response, without shutting off other things. And so the question is, what kind of cross-reactivity do we see in the population level? Let's zoom out and let's look at the good cross-reactivity. And we want to say, there's this study recently came out about how now that we've been a couple years past COVID, a lot of people have had vaccines and a lot of people have been actually been convalescent, have contracted COVID. And now we have antibodies to COVID from whichever direction. How much immunity does our population have against other coronaviruses? Look at the corona in the middle of this, orthocorona And so again, they have this tree. This tree is actually just a different version of what they had before. But you have all the SARS, and they even have Omicron and Delta here. And then they have a couple other brothers and sisters. But you see, once again, SARS-CoV-1 and SARS-CoV-2 are both pointing up. They're like at 11 o'clock and 10 o'clock on the clock. And then you have a bunch of other related things. You have MERS down at the bottom. You have the cold viruses. You have the OC43 and HKU1. Those are all the way, those are R and Q. Hopefully my head's not covering those up. Those are about 4 o'clock. And then those are actually also beta coronaviruses, is just the way they classify them. The other two cold strains are alpha coronaviruses. Those are located about 2 o'clock on the clock. Those are 229E and NL63. And then there's a couple of other more distantly related coronaviruses that are delta and gamma coronaviruses. So all that is to say, beta coronaviruses includes all the things we've been talking about and half of the cold coronaviruses. Alpha coronavirus includes slightly more distantly related the 2 other cold strains. And what they did is they tested to see, they compared pre-infected cells to, and they compared to see what kind of differences you got when you were actually infected with the SARS-CoV-2, and lo and behold, you have large antibody responses to the spike protein of SARS-CoV-2. Okay, that makes sense. And then the different, like the beta and Omicron, you also have large responses to those. Then they went down to SARS-CoV-1, still a large response. They went down to the other ones, all these weird coronaviruses, and you tend to have higher responses to all of these, except for this weird one, the Zhejiang 2013. That's a particular coronavirus that we don't have cross-reactivity to, but see that that's the exception rather than the norm. The easier thing to look at is to look at this where we look at pre-pandemic. We have all these coronaviruses, and the one thing to know, SARS-CoV-2 is all the way on the left on all these, and then you have the cold coronaviruses. There's 2 of them in the middle and 2 of them on the right. The pre-pandemic serum had responses to all of these cold viruses, but not too much else. Many of these other things like MERS is on the left side, Omicron, Beta, SARS-CoV-1, those are all the way on the left. You see it's completely flat on the left pre-pandemic. Then convalescent, they have the exact same order, but you see that, of course, the other, the stuff on the left has gone up. After you've convalesced, most people have antibodies at a population level. We have a lot of antibodies. They've gone up on the left for all these things. But there's also a lot of other things. Most other things they've gone up for as well. Not as much. It's a little bit of a dip. If you look between the far left, where you have the SARS-CoV-2 response, and the middle, where you have the cold response, you can compare the pre-pandemic and convalescent. And you can see that There's a few that you don't have cross-reactivity to, but the rule is cross-reactivity within these closely related coronaviruses. And if anything, you have more response to the cold viruses. They seem to be a little bit higher, although there's a lot of error in that. It's definitely not gone away. So immunity works. If you've had the mRNA vaccine twice, that's only spike. So the question is, are you responding? How well do you respond? Well, this looks like really good. You know, it's higher up on the left. The cold viruses are still high up, but the middle left, which is where you have all the related viruses, you have those all going up as well. Third time mRNA vaccine, well, again, it looks like you're getting better on the SARS-CoV-2 maybe, but it doesn't look like that much of a difference. And the cross-reactivity is not markedly increased. It's also not going away. It just looks like 2 times mRNA vaccine and 3 times mRNA vaccine is not that different by this one measure. Now, if you're infected and you have 2 times RNA vaccine, that's actually higher. And you see how this is actually, it's the same story. Infected plus 2 times, it's kind of like being exposed 3 times. But you actually have more cross-reactivity if you've been infected. The infected means you've been exposed to more epitopes. And in general, your immune response probably was turned up and focused in the right way a little bit more than if you just got the spike epitope. 3 times mRNA vaccine plus infected, not better, but not markedly worse. You might argue it's a little bit worse, but I think there's confounders. I wouldn't trust the data to say that it's definitely anything but the same. It looks the same. And that is the end of that. Now I'm going to go on to video 2.